DNA, transcription, translation, and mutations. Here we have a DNA sequence that should give us a 5 amino acid long protein chain. We read the DNA in the 3' to 5' direction indicated here. The top strand is our template strand, which contains the nucleotides that will be synthesized into mRNA. The coding strand will look like the transcribed mRNA, except all the thymines and DNA become uracils and mRNA. The mRNA strand is copied from the above coding strand in the 3' to 5' direction and is then rewritten in the 5' to 3' direction. Now we must translate the mRNA strand into the appropriate amino acid sequence. We look for the AUG codon to indicate the starting position. We continue in sections of 3 nucleotides until we hit the UAA codon, which is a stop codon. The stop codon does not have a linked amino acid. The 5 codons before, including the start codon, do have an associated amino acid, which creates the chain. This DNA sequence creates a methionine, valine, arginine, phenylalanine, and lysine amino acid chain. Here are 2 single nucleic acid point mutations. The first is a missense mutation, where we see a single nucleotide mutation causing the creation of a different amino acid. The DNA strand shows a series of 3 nucleotides that normally codes for valine, but in the mutated DNA, the guanine is replaced with a thymine showing a transversion mutation. This causes the mRNA to now code for the codon UUC, which produces a different amino acid, phenylalanine. The second box shows a nonsense mutation where a single nucleotide mutation changes what normally codes for an amino acid to now code for the sequence to stop being read earlier than it should. The first adenine and the 5th codon of the DNA coding strand is mutated and is now a thymine, showing a transversion from adenine to thymine. This causes the new mRNA to show a stop codon here rather than an amino acid, cutting the chain shorter. Here are 2 more single nucleic acid point mutations. The first box shows a silent mutation where a single nucleotide is replaced by another in a manner that allows for the same amino acid to be added to the chain. We see the 3rd cytosine on the 2nd codon here becomes a thymine on the mutated strand, showing a transition from cytosine to thymine. This now changes the mRNA sequence from GUC to GUU. Our codon chart shows that these 2 codon sequences result in the same amino acid being added. The second box shows a read-through mutation where a single nucleotide changes a stop codon so that it now results in an amino acid being added to the chain and possibly more after. In the coding strand, the thymine is seen to be replaced by an adenine and a transversion. This causes the mRNA to now translate a lysine amino acid rather than halt the translation process. These are the references.